Alma Carten, and I'm delighted to see this room so filled, and thank you all for coming to this most timely seminar on neuroscience and social work practice and social work implications for strength-based practice. You all know, as noted in the invitation, that social work more and more is drawing on these sciences to strengthen the knowledge base of our profession. And that's very consistent with the 12 challenges. Does anybody know about the 12 challenges? That's increasingly, I know you do, Dr. Lindsay, that's increasingly calling upon us to strengthen the scientific base of our profession. The 12 challenges actually are sponsored by three, and you can correct me if I'm wrong, because by three major social work organizations, NASW, CSWE, and the American Academy of Social Work and Social Welfare. So we really are being called upon to draw more on the sciences for social work. So I'm delighted that you're all here for what promises to be a very full session. We have an outstanding panel of members, outstanding professional panel with outstanding, you see their professional portfolios on your handout on your program. So we're very fortunate to have them all with us today. I want to introduce Gary Parker, who is the Deputy Director of the McSilver Institute, and Gary will serve as the moderator for our question and answer session. I also want to take the opportunity to give thanks to McSilver staff, Laura Morrison, and Amanda Alacantanda. They do an enormous amount of work that made this all possible. They do all the background work. I have a very small role to play, but I do have the distinct honor of introducing Mary McKay, Dr. Mary McKay, who is the director of McSilver. And I can tell you, Mary I admire a great deal. I don't know if you knew that. You probably figured it out. Mary has brought the McSilver a very long way in a very short period of time. So thank you, Mary, for all that you do. And please, Mary's wearing two hats tonight as McSilver director, and she's going to introduce our keynote speaker, and she's going to return to join the panel to talk about why it's important for us to integrate this into social work practice.
Thank you so much, Alma, and thank you very much for being such a good friend to the McSilver Institute for Poverty Policy and Research. I should tell you that Dr. Carten is on our advisory board and at the McSilver Institute, and which has such an important mission that I know Alma cares about, as do all of us here, to address the root causes and consequences of poverty through new knowledge and actionable research. And so Dr. Carten has been really a good friend, supporter, challenger to our institute, and so I'm incredibly grateful. She is also, maybe too humble to say, she is retiring after really decades of contributions to our field of social work. So if we could just thank Dr. Carten for her. So I have the privilege of introducing our keynote speaker, followed by Gary. We'll introduce our esteemed colleagues here at Silver. And Dr. Griffin, I have to say that Alma just said to me over and over again, you have to help him to come. You have to introduce him to our community. You have to introduce him to our colleagues. And so you have a big fan. I would hire her for any public relations that you have going on. So there we go. There's a little consulting thing that's going to happen right here. So let me introduce you to our keynote speaker. Dr. Griffin is an assistant professor of clinical psychiatry at the other university uptown, Columbia University, and attending psychiatrist at New York Presbyterian Hospital. That I admire both Columbia and New York Presbyterian a great deal. He's been working tirelessly to develop more novel and effective therapeutic interventions, and we're very lucky to have you today address our community, followed by my colleagues at Silver. So can you join me in welcoming Dr. Griffin? Thank you.
Thank you for coming. And I'm really excited today to share with you some recent findings from research in my laboratory on the molecular basis of drug addiction vulnerability. As Alma mentioned, in addition to telling you about my research, I have another broad goal, which is to describe a general framework for us to begin to understand some key principles of neurobiology that we can start applying to our practices. You know, I believe, and I'm sure Alma shares this vision, that not only does thinking about neurobiology help to enhance our medical and social interventions, but that as we begin to start talking about our work in the context of neurobiology, we'll start to foster better communication and better collaboration within the broad and sometimes disparate field of mental health research and treatment. So with that in mind, I have kind of two stages of my talk. During the first part of my talk, I'll describe the general framework that lays out some key principles of neurobiology. And then in the second part of my talk, I want to describe how we have used some of these principles to gain new insights into drug addiction vulnerability, insights which we believe will help to drive us towards better treatments, medical treatments, as well as to inform us in new ways to have social interventions. So this whole idea that all of us in social work or in psychiatry or in neuroscience, that we can one day use the language of neuroscience to bind our fields together might seem a little far-fetched at first glance, right? Because neuroscience, like the language of finance or like the language of psychoanalysis, can sometimes seem very insular and specific, right? Like you need to spend years training in it in order to understand it. But I don't think that that's necessarily true for neuroscience. What I think is that most people in the audience here are already familiar with certain concepts of biology and development that are critical to understanding how the brain works. In fact, if you've had the joy of watching a child grow and progress from an infant with their spastic movements to an infant who has continuous, seemingly purposefully destructive movements later on, then you've already start to grasp certain concepts that are important for understanding how the brain works. Let's review a few of those. Actually, funny story, a true story. On the day that I was actually on my way to give a lecture in Alma's class, this is back in October of last year, this is my son Xavier. He's 8 months old, and if you think that I'm using this metaphor because I want to show pretty pictures of him, you're right. Right, it's just true, I'm guilty. But, you know, on the day that I was actually on the way to give a talk to Alma's class, I picked him up, and he reached out and grabbed my glasses off. Right, it wasn't just an ordinary grab, right? It was one of those expert ninja move grabs. And he even gave me that ninja look that you see in this picture right here, like he'd been preparing it and training for decades, even though he was just 5 months old, right? And when you think about how does an infant move from these spastic movements to a controlled movement, that's actually principle number one for actually understanding the brain. And that's multimodal processing. I'm not sure if you can see it all the way from back there, and I'm sorry, it's a smaller screen, but I'll read it. Multimodal processing. A simple movement of the arm requires coordinated action of multiple muscle groups, right? In order to pull something back, in order to yank my glasses, his bicep had to contract, but his tricep had to relax. That process of activation of one region and inhibition of another region is how all of your muscles work. The higher complexity of the movement, like smiling, very difficult to make yourself do it because there's so many muscles involved. The higher the complexity, the more things that have to be activated and inhibited. This is how your brain works as well, multimodal processing. We'll get to that later. Another principle that's true for muscles, when we think about the brain also, is nature-nurture. So for my son, I can wish and hope and pray as much as I want, but it's very unlikely that he'll ever grow up to have the muscles of Arnold Schwarzenegger, right? It's just not going to happen because of my genes and his mother's genes. But it is true and very possible that with the right nutrition and the right training that he'll still have more muscular development or better coordination than either me or my wife. In the muscle cell, nature and nurture is where it interacts, at the level of the single cell, within the nucleus of the muscle cell. So every muscle cell is listening to see what type of nutrition you have, what type of activity you have, and then it develops and adapts accordingly. The same is true for the brain. Plasticity, because of this nature-nurture interaction at the level of the single cell, Plasticity controls how we treat muscle damage. For instance, if a person has a muscle damage, you can use an invasive treatment, but if you don't change the habits and the environment that led to the damage, you're wasting your time. Plasticity informs how we manage mental health because the environment and habits control how the brain adapts. And finally, development. That's the fourth principle that we should think about when we think about muscles, and it's also true for the brain. The risk for severe trauma, but also the capacity for rapid healing, is most high during the developmental periods. And we'll talk a little bit more about why that's true for both muscle and the brain. But it's a simple principle, all four of these that we're all familiar with. And we want to review how these are true for the brain. So how do these overlay onto the human brain? Your brain evolved over time to organize a very specific output in response to a specific behavioral input. And like the muscles, your brain is organized into modules, each with increasing complexity as you move from, sorry, I'm going to walk a little bit closer to here. As you move from the middle regions of the brain to the outer regions of the brain, the complexity of each layer increases. So for instance, the brainstem and the midbrain control things like arousal, sleep-wake behavior, blood pressure. We share these with all mammals. As you move up into the limbic system, this is where you see things controlled like fear, anxiety, emotional reactivity, reward-driven behavior. These we share with birds. We also share this region with reptiles, hence the common term reptilian brain. Finally, when you move out to the outer regions of the cortex, this is where things like abstract thought, but also inhibitory control over these other regions. Now, each of these regions develops in response to an environmental input. Molecular changes or cellular changes at the level of the single cell determine how these regions will respond to the environment. This level of plasticity varies from region to region, and it also varies by age. So if we look at the brainstem, midbrain, limbic system, and neocortex, as you increase in complexity of these different modules, the degree or capacity for each of these regions to adapt to the environment starts to go up. We can think of several evolutionary reasons why this may be true, right? Imagine your brainstem, which is controlling your breathing and your heart. You probably don't want that environmental change to affect your heartbeat too much. So that gets locked down pretty early, and we can't really affect it with behavioral therapies. But as you move farther out, the neocortex remains responsive, even in adults. I mentioned age. This here graph shows age moving, it's reversed, from 0 through 30. And as you can see here, the developmental window, these periods of high plasticity, opens and closes at different stages of development. Between ages 0 and 3, the midbrain is actually coming online. And actually between ages 3 and 15, this limbic system, which controls things like behaviors of fear, anxiety, reward-driven behavior, this is when it's most active and most plastic. So this idea that all of us share, that window of youth is the most promise to make changes and also at most risk if trauma happens, this is because these windows are open during that period between 3 and 15. Finally, the neocortex, this is what controls this impulse-type behavior, right? If you've ever had the feeling when you're talking to a teenager that they can't control their impulses and that they're not really listening to you, it's true. The neocortex actually does not come online or start to develop or fully develop until the age of 25. So a few implications for both the regional variation as well as the age-driven variation in plasticity are these. In the adult, most psychosocial interventions, or psychotherapies in fact, intervene at the level of the neocortex. This is called top-down influence. So if you take people who are doing psychotherapy and you do imaging studies, and this is still in its infancy, what we found is that the adaptations are often happening, or most often happening, in the neocortex. As we mentioned before, the limbic system, it's closed, it's done. It's already done its wiring between 3 and 15. Now, does that mean that there's no change in the limbic system from psychotherapy? No. But that change is because the neocortex changed and reached down and adapted to it. And we'll get back to that a little bit later because it's very important for addiction. A second implication is that in the adult, only highly salient events, environmental stimuli, can facilitate a change in the limbic system. These highly salient, meaning very important events such as trauma or cocaine, right? The brain is locked in, it's not really adapting anymore in this region, but there are events that can still change it. So PTSD can still happen, or cocaine addiction can still happen. But the talk therapy that you're doing, it's probably not talking to the limbic system. And this has very important implications for how we work with our patients. And finally, as I mentioned before, in the developing brain, these limbic regions are highly responsive to the environment. But the neocortex, it's not listening to you. So what's happening on the cellular basis? As I mentioned, each of these regions is mediated or changing because at the level of the single cell, there's a nature-nurture interaction. So what does that look like? Research that was conducted by my mentor, Eric Kandel, over the past 50 years, has shown that nature-nurture occurs to change synaptic formation in the brain. So in response to an environmental stimuli, the cell changes by growing new synapses, right? So if I start lifting weights, my muscles have the option of getting bigger. They might not, but that they have the option to, right? But your brain, because it exists within the finite volume of your skull, does not have the option of hypertrophy, right? So what the brain does, or what the cell does, is it grows new synapse formation. So the building block for cells, the building block for adaptation is new synapse formation, like leaves on a tree. And that synapse formation occurs in two steps. Activity outside the cell drives the signaling cascade into the nucleus, which is the control center of the cell. And signaling cascade, I'll use that term a little bit, probably pretty often in my talk. Think of it as a string of dominoes or a pool board. Molecule A hits molecule B, hits molecule C. But at the end of the day, it enters the control center of the cell. And the cell's nucleus responds by creating new proteins and causing new synaptic growth. That basic process is the centerpiece of adaptation within your brain. Two implications for that. A, nature-nurture, but the most important implication that I want to highlight here is that memory or adaptation is additive. The brain can grow new synapses, but it doesn't take them away. That means that once you've learned something or you've adapted to something, that adaptation doesn't go away. So we learn like rings on a tree, meaning you learn something at age 7, maybe it wasn't adaptive later, so you learn new inhibitory things at age 9, 12, 15, 4 months. But what that means is that the old memory never went away. It's still in the center of the tree. It means that each and every one of us has the capacity to act like an insolent teenager, and some of us more than others. Or perhaps maybe when you're sick, you have the flu, and you feel yourself regressing. That whole idea is that the rings on the tree are wearing down and the old you is still there. If you're treating a person that has addiction, maybe these synaptic growths have caused addiction, but then they get better 10 years, 20 years down the line. That addictive connection never went away, right? They were cured, but the addiction is still possible. It can always reemerge. So memory is always additive, never subtractive. We'll discuss that a little bit later. I mentioned the developing brain is a little bit different. What makes the developing brain for babies and infants, toddlers, and young adults different is that they have two mechanisms by which their neurons can learn. So shortly after birth, this here is a graph. This is showing time going this way. During childhood and adolescence, you have an increase, flowering of neuronal connections right after birth. So the brain creates multiple connections that don't have any relevance at first, but they create multiple connections, like leaves on a tree, shortly after birth. And those connections are there, and they get pruned away over time. So if we think about children as sponges, that's actually biologically sound, because they have so many connections that are sitting there waiting to see what you do. When activity is strong, a connection stays, but weaker activities causes pruning. It's the combination of activity-dependent pruning in the brain as well as activity-dependent gene expression that I described in the previous slide. The combination of those two creates these windows, developmental windows that we often refer to, that occur during the developmental period between infancy and age 22 or 23. So, how can we use this information to understand something like addiction So this is the second part of my talk. So as I mentioned earlier, I'm interested in studying addiction. So clinically, I see patients that have addictions to alcohol, nicotine, cocaine, sex addictions. And I'm interested in understanding what makes one person vulnerable and the next person resilient. Right? This has been a mystery in the field for eons, right? So if you give 100 people cocaine and you give it to them for 100 days, only 15% of them will become addicted to the drug. Only 15%. Right? Not what you'd expect looking at the media, but only 15%. Same is true for alcohol, same is true for heroin. 15% of people will get addicted. The other 85% can use the drug recreationally. This isn't a suggestion, this is telling you what the numbers show. And we want to understand what makes one person resilient and the other person not. Well, one of the first steps towards understanding what the risk factor is to look at the epidemiology, right? The population studies. What's different between the people that get addicted and the people that don't? You can look at things like prior use of other drugs like nicotine or alcohol, history of trauma, history of poverty, parental bonding, or other psychiatric, comorbid psychiatry like PTSD or depression. Of all of these factors, when you do the epidemiology, the two factors that stand out the most, that are most strongly correlated with subsequent illicit drug use, is prior use of nicotine or prior use of alcohol. In fact, this observation, often referred to as the gateway hypothesis, was first described in 1975 by my mentor, Denise Kendall, who's a professor in the School of Social Work at Columbia, that other school, right? And the gateway hypothesis is a population-based observation, right? And it describes a sequential pattern in drug abuse progression. Use of alcohol or use of nicotine typically precedes use of marijuana, which typically precedes use of an illicit drug like cocaine, right? And although the public policy debate always focuses on marijuana as the gateway drug, the epidemiology is clear that it's either nicotine or alcohol, individually or in combination with marijuana, that's a strong predictor of subsequent illicit drug use. But you know, in the 40 years since this hypothesis was first described, very few studies have really been able to tell us whether or not the order of drug use between the different drug classes, is it socially determined or is it biologically determined? Right? Is it because you know people who do cocaine that you go on to do cocaine, or is it actually that the use of alcohol or nicotine changes your brain? And if it is biologically determined, what are the mechanisms, the cellular mechanisms that mediate the process? We think that understanding or addressing these two questions can reveal new insights into the developmental trajectory of addiction. And that can also help us identify new molecular targets for treatment. So I'm going to pull a Rubio real quick. Okay. One way to address those questions is to translate the epidemiological observation into a rodent-based study that we can do in the laboratory, right? Translating the observation into a rodent-based study has several advantages. First of all, it allows us to address the gateway hypothesis in the absence, at least initially, in the absence of social factors that are already known to influence the development of addiction in humans, such as availability of drug. And then we can reinsert those social factors afterwards. Using a rodent-based study will allow us to vary the parameters of drug exposure to get new mechanistic insights. And importantly, because it's rodents, we can take their brains out and actually study what's going on inside. So we can test what's happening causally. For instance, we can use target-specific medical or pharmacological interventions, or even genetic perturbations in rodents where we change their genes to understand new mechanistic insights. So how do we translate this into a rodent-based study? It's not as easy as it sounds, right? In fact, as many of you already know, over the past 50 years, we've seen like a revolution in basic neuroscience, but translating that revolution into clinical or social interventions has been a little bit slower. You know, one of the reasons for the discrepancy in progress is the lack of adequate rodent models to use in our laboratory studies, right? You can model something like diabetes in a rodent, right? You can model something like even asthma, right? And do studies to understand what's going on. But how do you model something like depression in a mouse, right? Or what does a schizophrenic hallucination look like in a rat? It's very difficult, right? Even addiction is very difficult to study as well, right? Over the past 30 years, the gold standard for studying addiction in rodents has been cocaine or drug self-administration. This is an example, or just a picture of it, right? In this picture, right, the animal, this is a rat, he presses a little lever. The lever activates, activates the computer, which activates a pump, which injects a small dose of cocaine directly into his bloodstream. This port has been surgically attached directly into his heart. So in this model, the animal is able to choose when and how much cocaine he uses. Right? Amazing. It looks like addiction to me, but it's not addiction. Right? As we discussed earlier, simply using cocaine or liking cocaine is a big difference from being addicted to cocaine. 85%, 15%. The animal using cocaine is not addiction. So what I did when I was a resident in psychiatry at Columbia, I started collaborating with Eric Handel, my mentor, and I started trying to develop a rodent model of addiction that captures one of the key features of addiction, which is continued use despite negative consequence. So when a person walks in my office and says, "Doc, I'm using cocaine, I think I'm addicted, I don't know," one of the first questions is, what's going on in your life? How is it affecting you? What are the consequences? Right? Continued use despite negative consequences is a big fork in the road for understanding who's using the drug recreationally and who's using it addictively. So in this model, we actually insert consequence and force the animal into a conflict. So the animal does cocaine for 40 minutes, and there's no consequence at all for the first 40 minutes. Right? He presses the lever 5 times, and on the 5th lever press, he gets cocaine. So if you watch him in the cage, there's 1, 2, 3, 4, 5, then he kind of runs around, looks high. Then 2 minutes later, 1, 2, 3, 4, 5, he runs around, looks high. After the first 40 minutes, we start to insert consequence. During the first lever press, a blue light turns on. Wait, something different is happening here. Then he presses 2, 3, 4, and on the 4th lever press, he gets a mild foot shock. And I should specify this is a very mild foot shock if you've ever had the joy of putting your finger into a light bulb. Hopefully you have not, but it's a mild foot shock, but enough to make them change their behavior. If he makes it past the 4th foot shock and presses 5, he gets a cocaine injection. Each 40 minutes, we increase the severity of the foot shock. And the question is, how much aversive consequence is the animal willing to tolerate in order to get his cocaine? And our hypothesis, of course, right, is that animals with a prior history of alcohol use will have more compulsive cocaine use. Right? So to test that hypothesis, what we do is we have 2 groups. One group is just drinking water, and the other group is drinking alcohol for 2 hours per day. And then after 21 days of chronic alcohol use, we start cocaine self-administration, shown here in the red. Now importantly, the animals are only allowed to drink alcohol for 2 hours per day. We don't want alcohol in their bloodstream 24/7 because we don't want the cocaine and the alcohol to interact. So they do alcohol, then 18 hours later, at this time point, we start cocaine self-administration. And our question is, will the animals continue to use the drug despite negative consequence? As an added control, we ask, does prior chronic alcohol use enhance compulsive lever pressing for a sugar pellet? How does it affect the animal just for something like a natural reward? To do that, the model is very similar. 2 groups, one group drinks alcohol, the other drinks water, and then they start pressing the lever for sugar pellets instead of cocaine. We found, first off, that alcohol pretreatment does not enhance compulsive lever pressing for a natural reward like sugar pellets. As you can see here, this is 0 milliamps, 0.1 milliamps, 0.2, 0.3. With the mild foot shock, there's very little change in behavior. The animals will continue to press. They like the sugar pellet. They're taking it. No big deal. As you increase the severity of the foot shock, the amount of lever pressing that they do for the sugar pellet drops by 50%. And then by the time you get to a 0.3 foot shock, most animals have quit. We like the sugar. We don't like it that much. We'll wait till tomorrow. But as you can see here, having a history of alcohol use or history of water use made no difference at all. This is an important control because it shows us that the alcohol isn't just dulling the animals or making them unable to feel pain or making them unable to make a decision. So we know that the alcohol is not having non-specific effects on their fear circuits. When you look at how the animals who are doing cocaine respond, you see a different story. Animals who are alcohol naive, at the first sight of any type of consequence changed their behavior dramatically. They stopped pressing for cocaine by about 85%. And then as you increase the severity of the foot shock, they drop out of the study at 0.2 milliamps. Animals with a prior history of alcohol use, however, stay in the study much longer. With a mild foot shock, they're pressing up to 5 times more than the animals who are alcohol naive. And at a more severe foot shock, they're still in the study at 25%, down to 20% at the more severe foot shock. So there are two things that we can take away from this. First, it tells us that it substantiates our hypothesis, right? That a prior history of alcohol use does indeed enhance addiction-like behavior. It also tells us that the wiring to make this type of decision already exists for natural reward. And you can see how this curve is so remarkably different from this one, right? This is how a lot of people are, right? They might use cocaine, but when the first sign of pressure or negative consequence, miss a class, get pulled into the office, most people, 85%, will stop. But some people will continue. And we think that we might be on to understanding one of the risk factors that causes this. So we have here, actually for the first time, a full spectrum of the behavioral trajectory from a risk factor all the way to addiction. And we have this in a rodent model. And this puts us in a very good position to start asking other questions. For instance, what are the biological underpinnings of this process? And this is where we'll take advantage of some of the principles of neurobiology that I was describing earlier. So our first question in answering this question, how do you go from a prior history of alcohol use to being more susceptible to cocaine addiction? What region of the brain do we look at? Fortunately, we know already that certain regions of the brain mediate reward-driven behavior. For instance, this is a cross-section of the human brain, right, facing in that direction. In the context of a natural reward, it's a basic reward, like water when you're thirsty, a new love interest, or new food. Dopamine is released from the midbrain, this ventral tegmental area, to different regions of the brain that will then work in concert to help you remember both the sequences of behavior and the environmental cues that allowed you to get that reward in the first place, right? If I do this, this, and this, then she'll like me, right? That sequence of behavior. What we know is that in this region, the nucleus accumbens, this controls motivation. It controls your motivation to perform certain activities in the context of different environmental cues. Cells in the dorsal striatum, these control habit formation. This makes it so that you don't have to remember how to use chopsticks every time you eat sushi. And finally, cells in the prefrontal cortex. This controls, gives top-down control of both the striatum and the nucleus accumbens, right? This is inhibitory control. This is so you don't start eating sushi in front of a tiger, right? Or so that you don't start performing sequences of behavior when it's not appropriate, right? Each of these regions are important for regular natural reward. What cocaine, alcohol, nicotine, every drug of abuse has in common is that they increase dopamine in these regions, and they trick the brain. They trick the brain into thinking that it just found true love, or that it just found a new food source. And they continue to reinforce, whereas typically for a natural reward, you don't get that reinforcement over and over. And that's how addiction catches fire in these regions of the brain. Now, with addiction, most of what we know about is in the nucleus accumbens, or in this motivation center, and that's where I'll focus my talk. So if you look inside these cells, right, you take them out, crush them up, and look inside, you see that some of the same principles of adaptation that I was describing for muscle and neurons holds true in the nucleus accumbens. So if we take the nucleus accumbens, this is a nucleus accumbens cell, and we look inside, you see that in the context of cocaine or a drug of abuse, a signal transduction cascade, again, a bunch of dominoes falling over, sends a signal into the nucleus. The nucleus responds by creating new gene products, and those gene products go on to create new and stronger synaptic connections. The same adaptation process that we were describing earlier. Now for the past, well actually up until about 10 years ago, we thought that that was the end of the story, right? If you have the genes for cocaine addiction and you use cocaine, the signal transduction pathway happens and boom, you're gonna get addicted. We thought that that was the end of the story, but there's another layer of control that we've learned over the past 10 years that's very important. And this is called epigenetic, or above the genes, epigenetic control. And what it tells us is that the genes aren't just sitting there waiting for cocaine to come along to get addicted. The genes are actually tightly wound up around these balls called histones. They're tightly wound up in an inactive state. In that for them to be taken advantage of, so to speak, they need to be unwound in an inactive state. And there's two different, actually there's more than two, but we'll focus on two different classes of molecules that determine whether or not the genes will be available or unavailable. One group is called histone acetyltransferases. What they do is they add these negative charges shown here in red to the DNA and causes it to unwind and become available. And the other group of molecules are called histone deacetylases, HDACs. And what they do is they yank off the negative charges. So there's this constant battle between the histone deacetylases and the histone acetyltransferases. Most of what you'll hear about if you pull up a biology textbook or a random article is going to be about the HDACs, because there's a lot of studies studying these in cancer biology as well. So how does that affect something like addiction? Well, you remember earlier we were talking about my son, and you said, hey, well, he probably doesn't have the genes to look like Arnold Schwarzenegger, but with the proper nutrition and training, Jedi Master is still possible, right? It's these processes that determine whether or not his genes will respond to the environment. These two molecule sets are exquisitely sensitive to the environment. So they kind of do the gain or determine how your genes are going to respond. So how does this affect addiction? You probably can't read that. That says nicotine. But previous work that was done by Amir Levine in collaboration with Eric and Denise Kendall in our laboratory found that the other gateway drug, nicotine, actually acts within the neurons to inhibit one of these histone deacetylases. The consequence of that is that you get the DNA in this very unwound and very available setting. Right? So what he found is that nicotine inhibits HDAC activity, releasing the brakes on cocaine-induced gene expression. It creates a permissive environment. So with that mechanism, we were very interested in the possibility that nicotine and alcohol actually might have the same mechanism. So to test the question, does prior alcohol use enhance cocaine-induced gene expression of proteins that are involved in synapse formation, we gave the animals alcohol for 10 days in a row, and the other group was given water, and then we gave them just one test injection of cocaine. And we asked 3 questions. Does this type of exposure increase new gene expression? Does it add these little negatively charged things that are making the DNA relaxed? And does it do this by inhibiting HDAC activity? And what we found is that all 3 are true. As you look here, this is looking at new gene expression. Animals that are given cocaine, there's some gene expression in response to cocaine, for sure. But animals that are previously exposed to alcohol have enhanced cocaine-induced gene expression. If you look around the gene to see whether or not these negatively charged molecules have been added on, you see that prior exposure to alcohol causes a robust, almost 5-fold increase in these negative charges. So this tells us that alcohol alone is creating this permissive and vulnerable state within the neuron. And finally, it's doing so by inhibiting HDAC activity. So that's exciting, right? Because it tells us that HDAC activity is a similar process. Inhibition of HDAC activity is true for both alcohol and nicotine. So our next step is to figure out which HDAC. I had a little ball there that said HDAC. There's actually 11 of them. Right? Why do we want to know which one? Why go through all the trouble? Because what we're thinking here is that if we can figure out what specific molecule is the gatekeeper, then we actually have both a molecular marker to understand how other processes might make a person vulnerable to addiction, and it also gives us a molecule that we might be able to modify and treat in order to treat addiction. So through a series of experiments, we figured out that chronic alcohol use does this by inhibiting HDAC4. It's one of the 11 histone deacetylases. And what you're seeing here is 5 groups of animals. 4 of them had alcohol for 10 days, and we take their brains out at different time points after their last ingestion of alcohol. These animals, they were still drinking at the time that we took their brains out. And the other group, we took their brains out after the alcohol had digested and left their system. And what you can see here is that histone deacetylase 4 drops down precipitously after alcohol has left the system. What it tells us is that chronic alcohol use is causing a specific change in histone deacetylase 4. What that led us to believe is that histone deacetylase 4 might be acting as this node, right? Integrating environmental input to create a behavioral output, meaning potentiated or enhanced response to something like cocaine. But what we need to do in order to test that hypothesis is to directly inhibit HDAC4 in the absence of drugs, right? So a direct test of the role of histone deacetylase 4 as the gatekeeper. One way to do that is to expose the animals by exposing them to a molecule called MC1568, which directly inhibits histone deacetylase 4. This drug was created by a bunch of chemists specifically for this purpose. Again, I'm not the only person out there studying HDAC4. This molecule is very active and very important for multiple cells in the body. So what we did is we exposed animals to this molecule that specifically inhibits HDAC4, and we said, well, if this molecule is killing HDAC4, does that cause addiction? Does that increase the compulsivity that we saw? And that's what we saw. We saw that selective degradation of HDAC4 enhances compulsive cocaine use. So this here is first looking at the sugar pellet group. And you can see here that animals who've been given just vehicle saline and the other group given this drug, they don't change in their response to sugar pellets. Again, this shows that their brain is still intact, they can still make decisions, there's no nonspecific activity. But animals who are doing cocaine, you see a dramatic difference. The drug-naive group, no difference at 0.1 milliamps. But as you get to more severe foot shocks, the drug-naive group drops their behavior. They stop pressing for cocaine by 50%. And as you get to the more severe foot shock of 0.3, drug-naive animals are done. That's it. Whereas animals who are previously exposed to the drug stay in the game a lot longer. So this tells us or substantiates our hypothesis that not only does alcohol enhance cocaine addiction type behavior, that it's doing so by inhibition of HDAC4. There's two implications for this that I want to briefly review. First implication, as I mentioned earlier, if we can figure out how and why HDAC4, the mechanism, how it's being degraded, we might be on to understanding new molecules that could help treat the vulnerability to addiction. And indeed, we have found that HDAC4 is being inhibited by degradation. Chronic alcohol use is causing it to be eaten up by this, imagine a little Pac-Man running around in your cells, chewing things up. Everything gets killed eventually, but we found that chronic alcohol use causes HDAC4 to be inhibited. In fact, if you inject an inhibitor of Pac-Man, you actually rescue HDAC4. And that's exciting because it suggests that we might be able to prevent some of the addictive type behavior by injecting this inhibitor. The second implication is public health. We asked, well, what about the pattern of alcohol use? Does it matter how you drink? Right? If you're over 40 and you've been to a primary medical doctor, they probably said, hey, drink 2 glasses of wine. It's healthy for you. But if you went to college in America, you probably thought that, oh, that doesn't sound like a good idea. Right? Because college students drink very differently than older adults. And we wondered, is there a consequence to this? What we did is we tested groups of animals, one on water, one that drinks continuously, daily, the 2 glasses of wine a day, and the other group that drinks more like frat boys. And what we found is that an intermittent pattern of alcohol use does indeed degrade HDAC4 differently than continuous does. And it also causes more compulsive cocaine use. What I haven't talked about yet is some future directions, which is to understand what's happening in the adolescent brain. All of my studies are conducted with young adult to adult rats, because the adolescent rats, it's like a moving target. They have these short little windows. It's very hard to study, but we need to do it, right? Because the adolescent window is actually probably going to be more susceptible to this addictive process. We also want to study other risk factors, such as social isolation. I was talking with Patricia about that. So social isolation or PTSD. So in conclusion, animals with a history of chronic alcohol use do indeed have enhanced compulsive cocaine self-administration. Alcohol, like nicotine, inhibits HDAC activity in the nucleus accumbens, that is the reward center of the brain. It creates a permissive environment for this cellular adaptation that we were describing earlier. Alcohol causes HDAC4 to be inhibited by causing degradation of the molecule. And finally, and importantly, direct degradation of HDAC4 with a selective inhibitor, MC1568, recapitulates the entire addictive behavior, which shows us how important HDAC4 is to the process. And finally, just a few acknowledgments. In the Eric Kandel lab, several postdocs have worked with me, Philippe, Luca, and Kim Padu. I've been blessed to have several very talented research assistants. Peter's with me here right now, Peter Mercado. Previous assistants were Sal Yangli and Pragna. I have a lab technician who does the surgeries for the rats. That's Stacy Stevenson, Christine Doyle, lab manager, and also my mentors, Denise and Eric Kandel, who happen to be married. I forgot to point that out, by the way, not a coincidence. But they've been fantastic mentors, and that's it. Thank you very much.
I am learning a lot tonight.
Oh, I am too. That was
And I know I'm not alone in that. So my name is Gary Parker, and we're going to do Q&A, but I just want to hear from our other two presenters, and then we'll open it up to Q&A. And the great thing about moderating a Q&A is that I don't have to have any expertise in this area, and thankfully, because I don't, but I'm, again, learning an awful lot tonight. Thank you, Dr. Griffin. That was really wonderful. So I'll introduce them both. I'm not going to go into great detail. Their bios are in your program, and I want to make sure that we have an opportunity to hear from you tonight. So first, I want to, we have a great honor of having Dr. Judith Siegel with us. She's a member of the Silver School faculty, and she's published in countless different journals focusing on different issues, including intimacy and family relationships. And then we have Patricia Bryant-Reid, who is the Associate Executive Director from New Alternatives of Children, a child welfare, health, and social service agency. So now we're going to pull in the kind of social work emergence of this material, and we'll start with Dr. Siegel. So welcome her, please.
Well, Dr. Griffith, you're a hard act to follow. But this is exciting to be here, and I also wanted to thank Alma for inviting me. And Alma thought that rather than respond directly to Dr. Griffith, that I should really be able to take this into my own area of expertise, which is about the importance of family relationships. So what I'm going to do today, there's so many different facets of neuroscience and neurobiology, but I'm particularly interested in development and also attachment. So those are the things that I wanted to talk about today. And even though we're advertising this as a strengths-based approach, in order to really understand that and resilience, we need to understand the impact of trauma, especially on the developing brain. So that's where I'm going to be taking you tonight. And then ending with the concept of emotional regulation, and then in that way tying it into substance abuse. So a little bit of a social work with a family twist. So a lot of this is based on fMRI technology, which allowed people to question what was exposure to trauma during childhood. Did it change the brain? And they actually have developed all kinds of fMRI technology. So now they're able to look directly at a child's brain. But for the last 15 years, they've been looking at adults who were known to have trauma exposure during childhood and saying what was the consequence of that. So I'll start by introducing you to a concept that we all know about, which is just stress. When you are in a situation that is potentially dangerous, your body responds. This is something that, rather than it being top down, it's bottom up. Your amygdala, which is a part of the brain, is searching constantly to make sure that you're safe. And when you're in a potentially traumatic exposure or any kind of stressful event, your amygdala puts your body on alert. And so what we talk about is the activation of the HPA axis. If the construction vehicle that's outside of the window suddenly drops down and we hear a loud bang, not knowing what that is, even before our brain kind of tries to piece it together, our body is very excited. Cortisone, adrenaline. We are primed to either run or to fight or freeze. But when we talked about the reptilian brain, that's exactly what part of the brain is being activated, allowing you to survive. So when we say that these things are inbred, it's something that will happen genetically to ensure survival of the species, right? So it's a rapid shift in your body chemicals, right, that allow you to be on alert, and then this is intended for immediate response. You're not intended to stay in a state of high arousal for longer than a few minutes, right? So when we look at PTSD and we see someone, what goes on in someone who's on constant alert, right? What does that mean when we have a child whose cortisol levels are different when they awaken, right? These are children and adults who are constantly on alert. Their bodies are never able to fully relax and unwind. Well, what does that mean in terms of the brain? So I'm not a neurobiologist, neuropsychiatrist, but people who are and who have studied this talk about very, very important consequences to the developing brain. So when you look at children who are exposed to stress, trauma, and also children who are neglected, right, who are not given the levels of attention and soothing, there are important changes in the brain and the neurobiology. So as Dr. Griffith was explaining to us, that we have the potential for neural synapses to grow and strengthen, and the concept here is if you don't use it, you lose it. There's also neural sculpting. And these things become very important when we think about, well, how does the brain respond to being excessively and overstimulated at a very young age? Part of the things that they notice are reduction in the corpus callosum. I'm not going to get into any of that tonight, but when we talk about emotional regulation, it often is the neural networks that are connecting right and left brain functions that allow us to be thoughtful, to take learned experience, and to work that in with what the amygdala is activating, the state of high arousal. The corpus callosum is essential in connecting the left and the right brain. There's a concept of interoception, which is the ability to be aware of oneself. I asked a woman once who I was working with, are you aware that you're crying right now? She was very upset, and she had to touch the tears. She would not have known that she was crying unless her fingers showed that she... So this ability to know one's own self, to know one's own emotions, which are very important for emotional regulation, really depend on having this active connection between the right and the left brain. The disruptions in the endogenous opiate system are really essential. And so when we look at addiction, and the other notion of not just the reward center, but it's also questions about soothing. Are people using drugs as a self-medication? Are they trying to calm themselves down, right? So that's the alternative explanation that we need to ask about. Well, this ability to be comforted seems to be really related to that. And then finally, if we have this concept of synaptic pruning, are we able to look at that and look at what parts of the brain are changing and how does that affect things like judgment and impulse control? So these are, I'll let you read these slides if you're interested. You can get a copy of the PowerPoints, I think. But these are other studies that have looked at what are the changes like and how is that going to affect something like emotional regulation, which is just so important. This is from the work of Dr. Bruce Perry. Hands up if you're familiar with Dr. Bruce Perry. All right, so Bruce Perry is interested in the developmental age that trauma or neglect takes place and has developed a neurosequential model. But you can see, without being neuroscientists, that the brain of a 3-year-old who's been neglected is very different than a normal brain. I think particularly with the corpus callosum. But for Dr. Perry, that's really relevant to what kind of treatment, what caused this, but also what specifically can help. And it certainly isn't words or talking, right? So his sequential model builds on a very different perspective of the developing brain. So if we have to think then about trauma and we're adding neglect to that, as social workers, what is our mission? Both for intervention, but also for prevention, right? So we're very interested in saying, well, if neglect causes this damage, what populations are at high risk, and what can we as social workers do about that? So I wanted to talk a little bit about the exciting thing. Neurobiology isn't only about damage and what goes on in exposure to trauma. It's also about resilience. What predicts resilience? What allows us to understand the healthy brain, and what can we promote? And the answer has a lot to do with attachment. So attachment, again, is that very early, again, it's survival, it's built in. It's the ability for a young child, for an infant, to feel that there's an adult there who's available to comfort. And in attachment theory, they like the word attunement, right? To be able to read a state and to be able to manage a child's emotional dysregulation. Remember I said, if a child's in a dysregulated state for too long, that's already affecting the brain, all of that cortisone and adrenaline. How does a child get moved out of that state? Well, they get comforted. And they get comforted because their secure other is there to notice their distress and to help manage it. So there's a scaffolding, right? What an adult does, what the attachment figure does, is help that young child feel protected and comforted and eventually learn emotional regulation techniques. So, all of that becomes something that we have to say, well, if we know that, how are we able to translate that into intervention and, of course, in prevention? So what I wanted to do, if I can get this going, I don't know if any of you have seen this, but it's a wonderful, it captures the attunement between a child and a mother. And of course, he has a variety. If you want to YouTube this, you're going to see there's all kinds of cultures. There's a Korean mother and baby and a Chinese mother and baby. But what you're going to see is how important it is for a very young child to be able to read the parent and to be responded to. That's attunement. So I need help. I need someone who's a technician to...
He's doing it remotely. He's doing it remotely.
This young are extremely responsive to the emotions
Okay.
and reactivity and the social interaction that they...
Sorry, we're gonna open up the YouTube link.
You want me to do it?
Babies
Okay.
This young are extremely responsive to the emotions and reactivity and the social interaction that they get from the world around them. This is something that we started studying 34 years ago when people didn't think that infants could engage in social interaction. And the still face experiment that the mother did was she sits down and she's playing with her baby who's about a year of age. And she gives a greeting to the baby gives a greeting back to her. This baby starts pointing at different places in the world, and the mother's trying to engage her and play with her. They're working to coordinate their emotions and their intentions, what they want to do in the world. And that's really what the baby is used to. And then we ask the mother to not respond to the baby. The baby very quickly picks up on this, and then she uses all of her abilities to try and get the mother back. She smiles at the mother. She points because she's used to the mother looking where she points. The baby puts both hands up in front of her and says, "What's happening here?" good is that normal stuff that goes on that we all do with our kids. The bad is when something bad happens, but the infant can overcome it. After all, when you stop the still face, the mother and the baby start to play again. The ugly is when you don't give the child any chance to get back to the good. There's no reparation, and they're stuck.
And now I'm back to my PowerPoint. So, then, and we can talk about that in questions if you want, but that just shows you this attunement, right? That baby is attuned to mother's attunement to the baby, and that it's through that experience, if we're talking about synaptic connections, that self-soothing and comforting becomes something that the child is slowly able to do. So we have psychoanalytic theories from Kohut and from all people, different people about the importance of the other, of the object. But neurobiology goes, yep, it's all true. Babies need that kind of security in order to thrive. So I'm just going to show you some pictures that I like. But it starts very early, right? That infants solicit this kind of attention from us, from the mothers, and that this idea of the eye contact, of the ability to be known, to have attunement, is very, very important. The proximity and the security that comes from that attunement. But it doesn't stop there, right? It's something that throughout childhood, this idea of relational attachment, the relationship, which provides the security and the context for health. So, this is about our questioning when we're working with high-risk moms, when we're aware of it really from any families, and people like Sue Johnson are saying, even if you're looking at couples, this provides you with a new way of understanding health and ways to promote health. Now, when it's not there, let's look at what goes on. So your amygdala is startled, right? There's something happening. So we call that a trigger, talking about the common language that we're going to connect, right? So you get triggered, your adrenaline, cortisol is flowing. Now, if you don't have a good connection between the left and right brain, if you don't have the neurobiological basis to be able to manage your emotions, you can go right into behavior. You can go right into drinking more or smoking more. You can go into psychological depression and manifestations of stress. You can go into physical headaches and body aches, right? So this is all about the body and the mind are really one. So how are we able to understand that? If I say someone is dysregulated, that means that something has triggered them, and they do not have the ability to tolerate, comprehend, manage their emotions, right? So that's, and you all have known these people, whether they're in your office or at home waiting for you tonight, right? So something happens, and you can call that an overreaction, right? That here they are so violently reacting to something that in your mind or in many of our minds wouldn't necessarily have called for that. Just want to make you aware that that's not the only thing that can happen when we are dysregulated. We can implode. And the kind of thoughts and beliefs and emotional experience we have when we're imploding are just as damaging as the way that we act when we're exploding. And so when you're working with someone in the room and they're imploding, that becomes important for you to pay attention to and say that this, whatever's happening, isn't safe. We have shutdown, which Bowlby thought was the ultimate in helplessness and surrender, and Crystal, right? My DSW students. But when we're working with adolescents and young adults, we can see different forms of dysregulation. And I'm going to suggest, any of you here working with people who cut? Right? So that's when it's the me and the not me and trying to get myself back. Dissociation. Little ones can dissociate. There's symptom lists of how we can understand what dissociation looks like in a 2-year-old or in a 4-year-old or in a 6-year-old. When you're not able to manage the state that's been triggered, right, you leave it in one way or another. And this is one way or another. So when we very often see young children drinking and smoking because it's available to them, this is the other theory of addiction, which is I am escaping. I am using that drug to change how I am interacting with my environment right now, and whether it's an escape, right, that I'm changing the station. And when you get kids 14, 15, 16 years old who are going into drug rehab, they just didn't start. They started when they were 11 or 12. They start really young, and they keep on going. So these are examples of emotional dysregulation, and I just want to kind of tie this together now. So from a social work perspective, we're able to understand how exposure to trauma or stress, in the absence of that kind of secure attachment, can cause changes in the brain, which make it very, very difficult for a person to learn how to be emotionally regulated. And that in a dysregulated state, a person engages in behaviors or physical responses or addictive behaviors that are very problematic. So as social workers, we ask ourselves, well, what can we do about that? And here's where, if we have an exposure to a high-risk mom, there's a lot that we can do. We can engage her in finding ways not only to regulate her own self, right? So we have two of my esteemed colleagues who just received a grant to be able to work with high-risk moms who have PTSD and anxiety disorders. You know, how can we help a parent become regulated so they can provide that soothing and bonding to their child? But we have to engage these moms, and we're not going to do it through pathologizing. We have to find meaningful ways, whether it's in group or in an individual format, of helping them realize that we're here as a resource, that we really appreciate that parenting is extremely tough, even when they're as gorgeous as your son seems to be. And how are we going to be able to, on a larger scale, on a mental public health scale, really understand the impact of multiple stressors? So, you know, this whole idea of allostatic load, another language that we're going to build on, which is how can we help us prevent people from reacting so harshly to triggers when they've got so many multiple stressors? How are we going to be able to make interventions on their environment as we help work mainly with attachment and what we know creates a healthy brain? So that was what I wanted to share with you tonight.
Good evening. I'm actually going to talk, well, let me just say that when Dr. Carten asked me to participate on this panel discussion, I thought that the best way for me to contribute is to talk about my agency's transition into a more trauma-informed environment. I am one of the Associate Executive Directors at New Alternatives for Children, which is a health, child welfare, and social services agency. The clients that we service are families who are caring for children with medical complexity, children with developmental disabilities, and children with a range of behavioral health disorders. These families are families that are child welfare involved. So all of the families are referred because of some form of child maltreatment. So when they come to us, we see families, parents, and children who exhibit just a cluster of mental health disorders. As we began to become more trauma-informed, and as we began to look at these families and assess them through a trauma lens, it was very evident that many of the behaviors, many of the mental health disorders that we were seeing were very much related to trauma. So I'm going to show you the cluster of presenting problems that our families walk through our doors with. I think I turned it off. Okay. So these are some of the common presenting problems that our children present with. And many of them present with several of them at any given time. So we see a lot of aggression, we see a lot of emotional dysregulation, a lot of learning problems. Adolescents are often referred because of oppositional defiant disorders, conduct disorders. They're experiencing school-related problems. They're not learning. They're avoiding school. There's depression and anxiety. The parents, as stated earlier, many of them are being referred because of child maltreatment. So they're being referred for chronic neglect. So there's medical neglect, there's education neglect, there's inadequate guardianship, harsh parenting, physical abuse. And many of these parents experience or are presenting with untreated mental health disorders. So we see a lot of depression, a lot of anxiety, and comorbid conditions. Many of them are also not healthy physically. In addition to the mental health disorders that they also have a lot of untreated medical conditions. Sorry about that. All of the families that we're servicing live and are experiencing intense psychosocial stressors, significant psychosocial stressors. We have families that are having difficulty meeting unmet basic needs for food and clothing. At New Alternatives for Children, we have some flexible funding, so we have a food bank, a clothing bank, even a toy bank. Many of the children do not have toys. There's substandard housing. There's enormous stress because, as I stated earlier, in addition to all of the psychosocial stressors that they're experiencing, many of these families are caring for at least one child, and often two or three, that have complex medical disorders. And we're also seeing a lot of families that have at least two children with autism. So they are under an enormous amount of stress, which is further compounded by the fact that they have also come to the attention of the child welfare system. We are seeing a lot of undocumented immigrants, and as you know, they present with very unique problems because oftentimes because of their status, they delay in accessing treatment and services. And many of the families have poor or very conflict-laden social supports. So because of the high prevalence of trauma that we were seeing in the population that we work with, our mental health clinic, which is co-located within my agency, took the lead in trying to help us to learn as much about trauma, the impact of trauma, so that we could improve our assessment and our screening and assessments of these clients and our treatment. So they took the lead, and their interest in pursuing this information really coincided with just a wealth of literature and a wealth of training tools and training opportunities that we were able to take advantage of. So we developed a committee, a cross-section of staff from the different departments who wanted to work on helping us to transition into a trauma-informed environment. And many members of the committee also reached out to SAMHSA. And at the time that we reached out to them, we were able to benefit from the tail end of their outreach to community mental health agencies to provide training on trauma. So we received a series of rich, comprehensive trainings from trainers from SAMHSA who came on site and stayed with us for, I'd say we were the recipient of trainings from SAMHSA for about a good month, so that it enabled all of our staff. And I want to also say to you that we also included our support staff, because many times the people, our secretaries, our receptionists who are on the phone, they hear the distress from the parents. Oftentimes they are the first point of contact for many of the families that walk through the door. And they see and witness in our waiting room the emotional dysregulation of children, the harsh parenting that is oftentimes exhibited, and they have been impacted by experiencing the difficulties that our parents experience. So we received a lot of trauma. We also, our prevention program, we have one of the largest preventive specialized medical developmental disabilities prevention programs in the city. And so we were also able to have our prevention social workers participate in the Safe Mothers, Safe Children's project, which was a collaboration between ACS and NYU. So as a result of experiencing all of this training, we were then able to identify trainers within our agency, and they are responsible for ensuring that the training on trauma is institutionalized within my agency. I want to talk a little bit about how the training in trauma informed some of our treatment strategies. And one strategy that I'm going to talk about is very much related to Dr. Siegel's presentation. But before I talk about some of the strategies that were informed by the training that we received on trauma, I want to talk first about the strong emphasis that we placed on paying attention to the engagement process. The families that we serve are what we would call involuntary clients. They have not called us up to ask for help. Oftentimes they have been forced really to accept help. So we have to work really hard to engage them, to make them feel validated, respected, and safe. It's only when they feel safe with us will they then be open to any interventions that we would like to provide them with. So we use two strategies that I feel that are worth mentioning because they are very effective. We have found that they're very effective in working with this population. One strategy is the use of parent advocates. We have hired a group of parent advocates who themselves have had child welfare histories, who have experienced out-of-home placements, who were addicted. So we use them as a bridge between the social workers, the clinicians, and the parent. They provide, they share lived experiences with many of the clients that we service. And as a result, they provide a credibility. They are credible to the clients that we service. They share how they have been helped, how they have utilized mental health services, how they have worked to change behaviors and interactions with their children. And we have found that as a result of using these parent advocates, that the families become much more open to buying into the interventions that we are trying to provide them with. The other thing that we found that is very effective is that because these families are under enormous amount of stress, because they are experiencing just an enormous amount of psychosocial stressors, which are very much related to poverty, so that we really meet them where they are. You know, if they are having difficulty feeding their children, clothing their children, if they are at risk of homelessness, we have a flexible fund that we can tap that we use to really help meet some of their needs, which then allays some of the stress that they are experiencing, and which then makes them open to the interventions that we're trying to impart to them. I'd like to talk about one particular program that we have experienced a lot of success with, and that is a program called Building Blocks. This program, to me, is the one program that I feel has utilized the information on neuroscience and the neurobiology of trauma. This is a therapeutic program for young mothers who have histories of depression, trauma, addiction. Some of them may be cognitively impaired. Some of them may have been separated from their children because of out-of-home placement. Those mothers, so this program is specifically for those mothers and their young children between the ages of 0 to 4, and we also have some 5-year-olds. And the whole purpose of this program is to facilitate healthy attachment and bonding. So these parents and their children are engaged in this treatment, this dyadic treatment. It also, the therapy also includes a videotaping. So the families, the parents, when they come in, their interaction with their child is videotaped. And then each time that they have a session, the session is videotaped, and they are able, with the treatment, to actually see the improvement in the interaction between themselves and their babies, very much like the the video that you shared. They are really being taught attunement, attachment, and bonding. And this has been very well received. We have almost 100% participation from these parents. Again, I think that in terms of relating my experiences at the agency and with the discussions on neuroscience and the neurobiology of trauma, this particular program is very much informed by the knowledge around neuroscience. So that's one of the treatment modalities. Our clinicians have been trained in trauma-focused CBT. Parents are engaged in a parent journey parent training program. Parents also receive staged therapy where they're helped to identify their feelings, label them, identify triggers, before they're able, learn how to regulate their emotions before they are really helped to talk about and work through the trauma that they've experienced. And so let me just share with you a couple of positive outcomes that have occurred as a result of our agency becoming more trauma-informed. There is improved and meaningful engagement of our child welfare parents. Again, a very, very difficult population to engage. There's been a decrease in child maltreatment. We're able to close our preventive cases successfully. There's improved buy-in to the treatment interventions that we're trying to get them, help them to use and benefit from. There's strong attendance and participation to the Building Blocks program, parent journey that we provide. Families are safely being reunited after experiencing a foster care placement. And children who are not able to go home are successfully kept into their foster homes, who then end up adopting them. And we found, because we have better screening and assessment tools, that there is now a decrease in the psychotropic medication of children who have been misdiagnosed with ADHD, and even children under the age of 5, we've seen they've been misdiagnosed with autism.
So I do want to just end by saying the first of, I think, many thank yous to the panel tonight for sharing. Yeah, thank you. Yeah. And so I really think, in the interest of time this evening, I'm going to make my remarks incredibly brief to get us started, and then really turn to you as the active participants in this evening to ask questions of our speakers. So I do want to, though, thank Dr. Griffin, because Dr. Griffin, I've raised 5 children. They're now young adults. So thank you for sharing your child with us. I did, during adolescence, did not think that they were listening to me, and now I know that their brain was to blame, and that's good for me. But I gave them a very hard time, because at the time I didn't know that. So thank you very much. You know, I was thinking about your findings around tobacco and alcohol use, and some of the communities that we are so involved with in terms of community strengthening and mobilization, and access to liquor stores and exposures, selling cigarettes to young kids, and how, access to alcohol and tobacco and others, that varies by race, that varies by poverty. And so how much we invest in kind of those structural protections of kids that could help their brain, I think we're probably really missing that. And so I really appreciated your comments, and it gave me lots of other things to think about. I've heard Dr. Siegel talk a few times. Every time you get me with those pictures, Judy. And so, I really do want to thank you for underlying the importance of safety and security. That if children's brains are going to develop, they need to be in a relationship with a parent that can really tune into them. And I'm thinking about how many of the moms and the dads that we work with, that their life is so burdensome, that the ability to even sit and interact with their children are really incredibly important, and how much less we do investing in parenting support as we do in investing in systems after something's gone very wrong. And so you make that point beautifully in your slides. So then, Patricia Bryant-Reid, I had this image of new alternatives. I've never been to your agency, but by the time they got done with me, all I could imagine is folks walking in kind of with their brains like damaged and compromised, right? And you did such a really smart thing, which is that trauma-informed lens that you use greets people in a strength-based way, greets people with parent advocates, prioritize respect and safety and security first. And for so many of our child welfare mandated parents, they just don't experience those qualities of the services. So I think you did a beautiful job really articulating what a human service and a child welfare organization can really do to set the context for healing. So with that, I'm completely impressed. I've taken all kinds of notes, and I'm going to open it up to Gary to moderate our discussion, and to thank you once again for those wonderful talks. Thank you.
All right, we're going to jump right in. We're going to ask to keep our questions brief as well as our responses brief so that we can get to as many of you as possible, because I know there's a lot of folks here.
Great. I have a very loud voice. Oh. Thank you. I have 3 questions, one for each of the presenters, I hope. So the first question, okay, then I will do it in order. Have you experienced or experimented in the rats that you've collected having them reproduce? The control group who has no alcoholism and the alcoholic rats who do. And if in fact you haven't, I think it would be really exciting to do that. And my second very fast question is the effect of dopamine and growth hormone. There was a child who was diagnosed at 13 with growth hormone deficit, and at the same time was diagnosed with dopamine deficit. And the first time that young woman, after graduating college, did cocaine, forget it. She became a cocaine addict. So I always wondered whether part of the explanation of the biochemistry of the brain could be looking at different dopamine levels and different growth hormone levels. And my last comment is how proud I am to be in child welfare with your, not only do you provide child welfare services, but you do the whole self-actualization. You provide food, you provide rent, you provide toys. And I think that so few child welfare programs really think about that particular approach. Thank you so much, everybody.
Thank you. Some great questions. Dr. Griffin, you want to jump in?
Yeah, those are fantastic questions. To the first question regarding reproduction, I have not done those studies. It is on our list because what she's getting at is, do these risk factors transfer through the germline, right? And Yasmin Hurd over at Mount Sinai has found that animals who are exposed to marijuana, for instance, will have germline epigenetic changes that are transferred onto their children. So it has been shown, at least for marijuana, and we definitely want to see whether or not it's going to be true with alcohol and nicotine. Regarding the second question, as far as dopamine deficit, those types of disorders are one in, can't give you a number, but they're rare, what you're describing. Do they give us insight into what's happening with the addiction process? Absolutely, right? Dopamine is kind of the kickstart for this process, but in general, it's not happening in that way that you described, because with that type of disorder, it's so rare. But does changes in the ability of the receptors to understand dopamine affect addiction? Absolutely. There was an article in the New York Times just recently about this, by the way, and D1 receptors. Check it out.
Great, thank you.
Hello, I'm Jane McCall-Politi. I'm on the NGO Committee on Migration at the UN, dealing with migrant and refugee children's issues. So for a minute, I'm just going to go global. And first, for Dr. Griffin, it's a question about the developing fetus and the impact of the trauma that the woman is experiencing. I'm talking about refugee women trauma, either direct or indirect trauma. How, if there's a transgenerational transmission of that, and the effect on the child. At a conference, there was refugee youth, and they were, these kids really demonstrated a great deal of resilience. They had gone through all sorts of stuff. But they had had a good start, because I'm talking about Syria. The conflict in Syria started 5 years ago. The parents were educated. They had had the benefit of education. But now the 0 to 5 group doesn't have that base. And the refugee kids were saying, well, their mothers were too traumatized to parent. How do we deal with that? How do we protect the children? How do we protect their brains? How do we give them the benefit of the doubt so they can move on? Those are my easy questions.
Yeah, I'll skip that last question. That's a hard one. The first part of the question, I didn't point this out in my talk. That 15% that I was describing, it actually crosses socioeconomic, racial lines. So rich people, poor people, 15%, 15%, 15%. When you look at the opiate epidemic that's happening right now, we're talking upper middle class Caucasians, right? So this is not the kind of mindset that we had typically. It's crossing all barriers. So we're looking at something that everybody's susceptible to, which actually had me thinking about what you were showing, some of the subtleties in bonding. These are problems that I see in all of my patients, regardless of their background, some of these bonding problems. So addiction is kind of equal opportunity. The problems that lead up to it, the compensation hypothesis that she was describing, will some people be more likely to use the drugs earlier on because of other problems related to poverty? Then absolutely yes. But it is crossing all socioeconomic boundaries. And as far as fetal impairment, again, on the list of epidemiological risk factors, not as high as prior use of nicotine or alcohol, but certainly up there, and certainly can happen. But I would imagine that something like that is going to have more impact on emotional regulation, and then maybe downstream from that addiction. But I would imagine emotional bonding would be even more impaired.
Dr. Siegel, did you want to chime in?
No, I was just thinking that I was participating in a research study on the intergenerational effects of exposure to potentially traumatic events in China on a very large sample in Shanghai. And what we found was that the children, this is a group of children, I think we had over 2,000 age 5, were very much affected by the parental exposure, and even more so to the grandparents' exposure. So I think they're finding now that there is an epigenetic factor that children, they found it with the Holocaust children, that the children and grandchildren of Holocaust survivors. But a question that you're asking is, what can we do? So again, if we understand that there's a potential for trauma, then we have to respect plasticity and the potential for repair. And what we want to do is really provide all the opportunities, like you're doing with your agency, for families to be able to feel protected and to get the services that allow them to actually enjoy their lives in a different way at the same time as they're getting opportunities to bond and to create the attachment. So the brain is plastic, and so just as much as we need to keep that in mind too, that there's ample opportunities for these young children. It's never too late. There may be a better window, but it's never too late.
Ms. Bryant-Reid, I wanted to also ask to get your insight on this and working with different families and traumatized mothers. Are you seeing kind of an intergenerational impact?
Absolutely, absolutely. Many of the parents that we work with themselves have grown up in foster care. They have had losses and separations. As I stated earlier, it is really remarkable that many of them don't have a social network. And we know that if you don't have a support network, you're even more at risk of depression, anxiety, and all the other mental health issues, which then impacts and interferes with their ability to be attentive to their children's emotional needs.
What I'm finding so just fascinating about this discussion, it's this knowledge in action and its kind of practical implications right on the front line. So I thank you all so much for that.
Dr. Griffin, you said the finding was that prior nicotine and alcohol use led to addiction. Is there someone doing research on what leads to prior nicotine and alcohol use? No?
I think that yes, that's a very good question. I think we've touched on a few of them, right? Just, things like emotional dysregulation. There's experimentation, then there's continual use, right? So folks who are more likely to start and continue, we would start to ask questions about emotional dysregulation, but we certainly know that there are other social factors, such as poverty, or such as availability. Is there a liquor store on every corner? Are people selling the cigarettes? Is it available? So those factors would come into play. I also wanted to go back to the transgenerational thing one more time. The concept that certain behaviors can be crossed transgenerationally, there's evolutionary support for that, right? In the sense that if you do not have resources, right, or you live in an area, an animal lives in an animal that doesn't have resources, the liver, the brain needs to be ready for that transgenerationally. So there are epigenetic markers that cross the genes, that go into the testes and ovaries, that tell the next generation what type of environment you're going to live in. So stressful environments, can that be transferred transgenerationally? Absolutely. Whether or not addiction specifically can, we're not sure yet. But the idea that anxiety disorders, depression, these things are passed through the brain as well.
I just wanted to comment on the intergenerational things as well. When Patricia and I would have lots of discussions, we were getting really concerned that the problems that clients were bringing were so problematic, they were so complex, they were so compound that it always seemed impossible to treat them. And I recall, you may remember you're saying this to me, Patricia, but one thing you did say is that you found that the symptoms that the parents were bringing to you were like, I think you used the word masquerading for untreated trauma from the past. And I think that we as social workers, we always say if you don't do a comprehensive, broad-based assessment and getting the assessment right, you're going to get the intervention wrong and you're not going to be helpful to the client. So I think as we move into this trauma-informed care, it gives us a broader base for making comprehensive biopsychosocial that is informed by the new neuroscience. And I also think, and I don't want to sound very sophomoric as a social worker, but we get that way sometimes. But I think there's always the possibility of change, and I think behavior cognitive therapy is one way of doing that. When we begin to move away from what Professor Siegel said, from pathologizing parents to using interventions that work, and they may not be perfectly attuned parents, but they can be better attuned parents. And that's what I think about bringing this sort of strength perspective to the work that we do.
And I just want to say that I want to emphasize that we see an enormous amount of resilience. So, I mean, our families have complex issues and needs, but once I've seen really remarkable change, once they are engaged, once they feel respected and validated and safe. The parents have to feel safe in order to be open to the interventions, and I have seen that occur.
I just wanted to ask a question about there. You said there were a lot of benefits to this treatment. Were they able to reestablish a sense of attunement as a child?
There were parents who had been separated from their children from child maltreatment. As a result of that treatment and just other interventions, those parents and children were successfully reunited.
Hi. How are resilience or protective factors studied in an animal model if we're looking at something like addiction? Strengths-based, right? That's the whole point.
So it depends, resilience to what you're referring to.
So, or any sort of protective factor. So if we were to look at, for instance, the risk factor of having alcohol beforehand, let's say something like that.
Yes.
Or isolation, removing that as compared to a baseline of not having that stress factor.
Yeah.
What about the things that promote resilience against
Absolutely. Yeah.
risk? So maybe more social interaction or things like that. How do you really test that in an animal model?
So you can test it with rats because they're very social. And, typically we do our studies where the rats are individually housed because if you put them together, they help each other take off their equipment and it's a whole big mess, right? But it has been found that rats that are paired and socially grouped, they're actually more resilient against addiction. So pairing does matter. Social factors does actually make them more resilient. It's also been shown that rats that are separated from their mother early, or maternal separation early, makes them more vulnerable, whereas pairing them closer makes them more resilient. So it's also been shown that, I don't know if you've read these articles about the rat park, right? So rats that have an enriched environment, little things to play around with and little toys, are also more resilient against cocaine use than others. Again, most of those studies have unfortunately used that older model of substance use, meaning how much cocaine do they use or not. But I'm very interested in testing some of those theories, pairing, rat park, all of those things in the model that I use now, which is actually looking at something more clinically related to addiction. And that way we can ask that question a little bit more directly. But the rats, they like to hang out and connect and climb on top of each other. So there's lots of flexibility to do those studies.
Well, anybody that's ridden New York City's subway system knows all about the resilience of rats.
They eat pizza, right?
They see it in action every day. Do you have any? Yes.
So, having worked with rats and humans, is a rat a legitimate species to test addiction, especially considering, you mentioned the top-down processes that occur. So, what about going to non-human primates rather than rats? Because I think we've kind of reached the ceilings on rat addiction research.
The second part there are limitations, right? As far as reaching the ceiling, no, I don't think so, right? Because the ceiling was reached because we weren't using a model that captured addiction. We weren't actually examining these top-down processes, right? If you don't force the rat into a conflict, you don't engage those top-down processes. But are there limits to those top-down processes? Certainly, right? With humans, the standard is a 12-step model, right? Where you go into AA, the higher power. There's no higher power for the rats, presumably, right? So there's not a way to do the studies for those types of psychosocial interventions. Probably can't do it with the primate either. But yeah, there are going to be limitations. But as far as the rat as a model, one slide that I didn't show, or actually I didn't mention it though, the reptilian brain, it is so well conserved between humans, reptiles, mice, and apes, so well conserved that the architecture of those cells very, very similar. So it actually is a good model for studying that region. But the neocortex, right, we're so far evolved from rats that there's no way that we can really model the degree of complexity of what's going on with humans. So it will reach a ceiling, I think, for sure.
Dr. Carten, I want to offer you closing comments since you're the one that brought us all together, and I know we're so very grateful for the opportunity to explore this issue.
I really have I'm sort of always at a loss that there's nothing further for me to say, and those who know me know that I always have a lot to say. I love having the last word, but I think from your reaction, like I'm saying, but from your contributions to the questions, thank you very much. There's obviously you got a lot out of this. I can only say to Mary and the McSilver people, Gary, thank you for bringing this, allowing me to bring this to our silver community, and I can tell from the response that it was very meaningful for you. There was a lot of information here, an incredible lot of information, and I always know that the more I think I know, the more there is to learn. I also want to say that it's, for me, it's really wonderful to bring, I think as our profession moves more toward integrating science, we also have to move more towards an interdisciplinary approach to our work. And so I can envision classrooms. I kept saying to Dr. Griffin that we need to do co-teaching, because every time you were lecturing my class, I kept butting in because there was so much, there were so many similarities between clinical social work practice and your work as a neuroscience. So thank you all. This has been very fulfilling for me. Thank you, Mary. Thank you, Gary.
Once again, a nice round of applause for our panelists. Thank you for your wonderful questions. And please keep an eye on your email. We hope to see you at the next panel we have.